It's my pleasure to introduce the first speaker, who is Dr. Kensei Nishida. Professor Kensei Nishida is an associate professor at the Institute of Biomedical Sciences at Tokushima University Graduate School in Japan. He has authored more than 50 original papers and has had several review articles to his credit. His primary research focuses on the dynamic relationship between stress and gut-brain axis, and he is practicing internal medicine physician and researcher specializing in stress biology. It's my pleasure to invite Professor Kensei Nishida to talk about some aspects of the gut-brain axis.
Thank you very much for your kind. Yeah, so. No. This one, yeah. Okay. Thank you very much for introducing me. So first of all, I appreciate the president of the symposium, Professor Hungry, so for giving me such a wonderful opportunity to talk to you. So yeah, my name is Kensei Nishida from Tokushima University. So yeah, actually I'll speak today from a perspective of a researcher, but I have another title in the university. One is a gastroenterologist, and second is a medical educator for medical student. So I'm now little nervous. So okay, and the title of this presentation is a gut-brain axis and the probiotics and their impact on the health and unbelieving effect of sleep improvement and stress alleviation. These are contents of today's presentation. The first, I'll talk about the brain-gut microbiota axis. So yeah, as you know, that this concept has been well accepted in the not scientific community, but also the public, such as TV or the magazine or the internet news. And so I'll present a short review about this concept. And the second, so I will introduce some data of our clinical studies. And in this study, we have a very interesting function of probiotics. And so I share some data with you today. And first, this is my favorite words, what Hippocrates said. He said, this is the boils and the bad digestion is the root of evil. And he also said, all disease begin in the gut. If a disease cannot be cured by food, even a doctor cannot cure it. How can a person who doesn't know about food understand the diseases? So recently, so many studies about gut microbiota or gut environment has been developed. So we can easily renew the recognition of the importance of gut. But so it's surprisingly, so in 400 BC, Hippocrates also already mentions the importance of a gut environment for health. So yeah, as you know, the gut-intestinal tract has three important functions. One is the enteric nervous system, and the other, another two is the immune system, and the third is the endocrine system. So you know, so there is a huge neuron cells in the gut as much as those in the spinal cord. And also the 70% of immune cells of whole body are located in the gut. And more than 20 types of endocrine cells are expressed in the epithelial cells or submucosal cell layers. And gut microbiota, which is separated by a single epithelial layer, here, and it interacts with these three systems and consequently to regulate the gut environment. So the gut environment is controlled by hormonal and neural and immune interaction. So this information is transmitted to the brain through the vagus nervus or the blood flow. And actually, so 90% of the vagus nervus is afferent pathway. So here, and so indicating that the importance of the gut. So, when we get in physical and mental conditions, the balance of the gut microbiota is called symbiosis. And the imbalance of the gut microbiota, which is called dysbiosis, often leads to physical and mental disorders and various diseases. And many research have revealed that the gut microbiota is deeply involved in intra-organ networks such as brain, skin, bone, and so many organs. And these interactions often affect the pathological condition and the diseases as listed here. So the gut microbiota has an important role in homeostasis in our health via the gut-brain axis, as well as the other organ network. And to keeping a good gut environment in good condition is a key for our health. So, the probiotics has an improving effect on gut microbiota. So we want to clarify how probiotics can affect the gut-brain axis. So we designed the clinical studies and to design the human stress model and conducted the clinical studies. So in this study, we found the multi-potential effect of probiotics, such as stress relieving and improving sleep quality. So following slide, I will show some data about clinical studies. So this is our clinical study here. And the subject were a fourth-year student, the medical student who were taking a nationwide examination for academic advancement. And the fermented milk with Lactobacillus shirota strain, so now we call it LCS, and the placebo without LCS were used as a test beverages. And 8 weeks before, so can I see this? There, 8 weeks, sorry, back. 8 weeks before the examination, the subject started to drink the test beverage, and they were required to drink every day. And the stress changes, stress-induced changes were assessed by the means of by questionnaires and the salivary cortisol and the gene expression profiling on the whole blood, and also the microbiota analysis. And this is a flow chart of the background. And the total entry number is 172 students, and the final target is 70 students to both group. And here, right side is a background. So to assess the psychological status, we use the GHQ, General Healthcare questionnaires, and the STAI means state trait anxiety inventory. And all parameters are within the average levels in the same age groups. We confirm there's no difference between two groups. And here is the data about the symptom assessed by questionnaires. And this graph shows that rate of the expression of symptom. And white squares means placebo, and the black square is LCS. So left panel shows that, so here, and the abdominal symptom data. So you can see the LCS inhibited the appearance of abdominal symptom in prior 2 weeks period before the examination. And the middle part panel shows that data about physical symptom, so which include all symptoms as anything wrong, and such as the headache, the stomach ache, and the backache, and anything. And so you can see the placebo group increased the appearance of the physical symptom as the examination approached. However, the LCS suppress this increase. And also the LCS group and suppress or tend to suppress the frequency of cold symptom. You can see the right panel show. And so the participant taking LCS probiotics reduced the appearance of physical symptom during stressful condition. And this is the assessment of the subjective and objective assessment. And left panel shows that the experience of stress assessment. So to assess the stress assessment subjectively, so the participant were asked to the degree of stress on the visual analog scales. And they check the extent how they feel stressed on a 10 centimeter lines like here. And the left panel shows that you can see the red line, and LCS maintained a lower expression during the drinking period. And the right panel shows to assess the stress objectively. So we use the salivary cortisol levels. And it's well known as stress biomarkers. So you can see the placebo increase the salivary cortisol levels and maximum point 1 day before examination. However, LCS group suppressed the increase of salivary cortisol level 1 day before examination. And next, here we analyze the change of gene expression in the whole blood by microarray. And left panel shows that the expression of gene, the food expression changed more than twofold compared to the previous period. And left panel is the placebo, and here is the LCS. So you look with the light panels. And so that panel shows a number of the differentially expressed genes. And compared to the previous interpretation period. And you can see in the blue box and the placebo, the number of the changes genes were, so yeah, 160 genes were changed in the placebo before the examination period. However, at this point, the increase also suppressed in the LCS group. So, so this pattern is very similar to those in the salivary cortisol levels. So indicating that the LCS intake had a moderate effect on the physical stress response. So under the very, very stressful condition and such a 1 day before examination. Maybe you can imagine when you did 1 day before examination. And so this is an analysis of microbiota. So I showed the alpha diversity in the observed species. So LCS group maintained the higher levels of observed species in just before the examination. So suggesting that the LCS also improves gut environment. So in this study, so probiotics improve the gut environment and act protectively against the stressful conditions, the stress. And so the question is, does the gut microbiota really influence the stress response pathway? So are there any previous reported? So the answer is yes. So here is a very often cited data reported by Dr. Sudo in 2004. These graphs show that the stress response and HCTH corticosteroid, which means the HPA reaction, under the restraint stress mice model. So you can see the red circle. And the germ-free mice showed an exaggerated stress response compared to the SPF, the normal mice. And furthermore, so when the normal fecal was destroyed to germ-free mouse by 6 weeks of age, the HCTH stress response is recovered. And so here is a point, sorry. Okay, yeah, Yeah, okay, yeah, thank you. Yeah, this point, yeah. And however, after 8 weeks of age restoration, so the response of ACTH was not recovered, no more recovery. So this indicates that there is the existence of a limited window to normalize HPA reaction in germ-free mice after restoration of microbiota. So that it suggests that post-natural gut environment plays an important role in developing the HPA stress response pathway. So let us look at the relation between the gut and the brain from two aspects. And first, so from the evolutionary perspective, so very primitive, primitive jellyfish had only nerve that correspond to the enteric nervous system in human. And this indicates the nerves so involved in absorption and excretion of nutrition are the most important for organisms to survive. And with evolution, and the gut environment can be intricately controlled by ganglionic or spinal reflexes. And in mammalian, so gut information is transmitted to the cognitive brain, emotional brain, and integrated the hypothalamus, and again, and transmitted to the periphery. So from this point of view, the brain can be seen as derived from enteric nervous system, right? And the other aspect from the individual development, and the enteric nervous system is derived primarily from the vagal neural crest. In this, neural cells is migrating through the enteric tract and forming the enteric nervous plexus. So from point of view, so the enteric nervous can be regarded as a part of a brain. So anyway, so we can easily accept the idea that gut and brain are closely related. As you know, the gut microbiota composition changed changed over the course in the lifespan, and it affected many various life events from the birth to death. So especially during infancy, the composition of gut microbiota dramatically changed. So this is exactly times when the neural network is developing and some psychological disorders such as autism became apparent. So the researchers naturally suspect the relationship between the autism and the gut microbiota, you know? And so indeed, in 2013, the Cell journals, it is reported the gut microbiota can affect the host behavior, and the gut microbiota has come to be called the mind-altering microorganism. So here is the report about the link between the autism and gut microbiota. And so it is reported that the transplantation of the autism children, the fecal from the autism were transplanted to germ-free mice. The mice developed the symptom similar to the autism. And also clinical study has reported that the transplantation in the autism children improved their autism symptom. And this improvement has remained at least 2 years, for 2 years. So yeah, like these reports, so you know, the many researchers in various fields try to develop a strategy as a treatment using gut microbiota. So with this background and our clinical data, so we further investigate another more effect on the central nervous system. And then, so we focus on sleep. And you know, the insufficient sleep has been linked to some chronic disease and the condition as obesity, cardiovascular disease, diabetes, or depression. And here is the data about, this figure shows the sleep duration. So left panel shows about the area. So yeah, Japan is drawn red. So it means Japanese is the shortest sleeper all over the world. In addition, the people in the Asia, so also in the India, are the relatively shorter sleeper compared to the European people. And so sleep deprivation had a lot of a negative effect on the physical and the psychological condition on our daily life. And sometimes feeling irritable or angry or feeling overwhelmed, lacking interest, motivation, and energy, and so on. So insufficient sleep, so exaggerates the stress feeling. And also, in the same time, the stress induce the low quality of sleep. So the stress and the sleep can affect each other. And this is the mechanism of sleep. And here, stress environment is known to be one of the precipitating factor. And here is the flip-flop model to regulate the sleep as proposed. And this mechanism, there are two centers, sleep center and the arousal center. And in the daytime, arousal center is activated. In the night, the sleep center is activated. In the next morning, again, the arousal center is activated. Switching the center in the right time is needed for good sleep and good wake. And in this mechanism, orexin has an important role in activating the arousal center and also the sympathetic nervous system. Orexin is known to the neuropeptide produced in the hypothalamus, and orexin itself activated by stress. So the hypothesis here, and the stress-induced orexin may affect arousal center activity and induce the low quality of stress. And so when we think about the sleep, so we usually divided sleep two phases. One is a non-REM sleep and the other is REM sleep. The bottom is the sleep status. So non-REM sleep is deep sleep, N3 sleep, and REM sleep is relatively shallow sleep. So during non-REM sleep, the growth hormone is secreted and also the autonomic nervous system is switched. So it is known that the non-REM sleep is very closely related to the sleep quality. So in our clinical studies, so we focus the non-REM sleep in especially the first cycle here. And this is the experiment design for the sleep assessment. So the protocol is similar to the first one, and the same beverages were used. And to assess the sleep quality, we use the sleep questionnaires and electroencephalogram. And this is the flow chart in the background. Okay. And the final target is 46 student and placebo and 48 student in the LCS. To assess the basic sleep status, we use the PSQI, Pittsburgh Sleep Quality Index. It is well known as a sleep questionnaires. And we confirmed there is no person who is suffering the sleep disorder or insomnia. And also there's no difference between two groups in these parameters. And here is the questionnaires, which called the OSA, Oguri, Shirakawa, Azumi, the developers' names. And this survey consists of a total of 16 items from factor 1 to 5 and evaluates the feeling of sleep, and which fluctuates from day to day. And so we can calculate the total score by these 16 items. We also calculation the scores with when we did the stratified analysis, each factors. So I will show the factor 1, sleep on rising, and factor 5, experience of sleep duration data after this slide. First, so this is the data about anxiety and the total sleep scores. So you can see that all participants increased anxiety one day as the examination approached, and one day before examination point is a maximum point. And also the total sleep score worsened one day before examination. So subjective sleep status correspondingly worsened as an increasing anxiety levels. So, but there is no significant difference between placebo group and LCS group. So we did the straightforward analysis and we found this data. The scores on sleepiness rising and experience sleep duration maintained relatively higher in the placebo group throughout the study. So black is LCS group here. And yeah, this score worsened just before examination, and after the examination, the score is recovered. Yeah, the LCS maintains these scores. And next, to assess the sleep quality objectively, so we use the sleep electroencephalogram measurement. So as shown in these left pictures, the distance instrument had the advantage that the subject, I mean the participant, can be monitored at home, and because it is very small and easy to use. And from EEG data, so we can obtain these 6 items: sleep latency, and N3 sleep. It means the deep sleep time, and delta power during the first cycle. So delta power is considered as the ability to represent the ability to sleep deeply, strongly. And total sleep time and sleep efficiency wake after sleep onset. So following slide, we show that the yellow items 3 here. And the sleep latency prolonged in the placebo group as the examination approached. However, this prolongation was prohibited, prevented, and LCS group here. And so the duration of non-REM sleep decreased in the placebo group. Even after the examination, it's kept decreasing here, whereas its duration in the LCS group maintained higher levels, consistent higher level. And also the delta power of strength, so delta power in the LCS increased before the examination, and it remained unchanged in the placebo group. So this data suggested the person who drinking in a placebo LCS could sleep rapidly and deeply and strongly before the examination. These data suggested and support the improvement of the subjective assessment of sleep on sleepiness arising and experience sleep duration. So there are not so much report now about the link of the sleep and microbiota. However, some reports show that sleep deprivation affect the microbiota and also vice versa. And also the alteration of microbiota by probiotics can change the sleep pattern. That is rodent models. And so microbiota and epithelial cells, gut epithelial cells, has a circadian rhythm, and it is not surprisingly if probiotics can affect our circadian rhythm through the alternation of gut microbiota. So we have successfully found that probiotics have a stress relieving effect and the improving sleep quality effect through the gut, improving the gut environment. But so the mechanism by which the probiotics exert this function is not fully understood. And probiotics as one of the key items for strategy to develop and the gut microbiota strategies for health promoting. And probiotics had advantage that to be the food product, which means the safety and the tasty. Sorry. So this is the final slide. So while we are doing the clinical study, so we have to come to believe that improving sleep quality is an essential function of probiotics on our health. So in taking probiotics allows our body to recover from the stressful condition during the sleep and protect the physical and psychological condition from excessive stress response. So in other words, the probiotics enhance the stress resilience. So in terms of improving sleep quality, so the probiotics is expected to have other positive impact on our body, such as here. And yeah, that's 1 minute. So yeah, Hippocrates said, this is the boil and bad digestion is the root of all evil. So we can say that to keep a good intestinal environment with probiotics is the royal road to overcoming all evil and bringing health and longevity. And this clinical study were collaborated with Yakult Central Institute, Japan. So we all thank our collaborators and also the participants in the clinical studies. Thank you very much. Thank you for your kind attention.
Yeah, thank you, Dr. Nishida, for that wonderful lecture. We will have a special session at the end of this session for question and answers.
Okay.
So please join me once again in thanking Professor Nishida for a wonderful talk.
Yeah, thank you very much.
So I'll